looking at cell cycle regulation. So essentially, all the data that I'm going to show and talk about in the session relates to an advance that was provided in a paper published in Cell in 2008, which and is known as the Future Reporter. So the idea is that this team of investigators engineered a reporter such that when cells are in phase G1 of the cell cycle, then they express a red fluorescent protein. When they move into SG2M, they start expressing a green fluorescent protein. And then you get some slight overlap between G1 and S where things look yellow. And immediately after mitosis, they go dark for a short period of time. But apart from those sort of subtleties, the kind of really nice thing is just being able to detect essentially the stage of the cell cycle that a given cell is in. And one of the nice figures from this paper is on the right-hand side, where you can see very clearly that they can put this reporter into lots of cell lines or into lots of different organs and really be able to visualize the patterns in the cell cycle over time. And so we have been interested for a while in sort of thinking about these kinds of data and asking questions about if we can build models that incorporate these cell cycle dynamics, what can we learn about how the cell cycle might be regulated, and in particular through cell-cell crowding. So the real focus of the talk today is going to be all about what we can do in that context. To say things right from the start, I'm only going to talk about PDE models today. They're all going to be models that describe how the cell density evolves in time. And typically, I'm going to break my models down into effectively density of red cells, so they are cells that are in G1, and density of green cells, cells that are in SG2M. And I apologize from the top. There's a little bit of inconsistency in notation because sometimes I'm going to call them R and G, and sometimes I'm going to call them populations 1 and 2, but I'll try and highlight that. I'm interested oftentimes in parameter inference using sort of the data that I've got and these models that I develop, and that's mostly going to be in the context of a Bayesian framework. So I'm interested in understanding whether we can estimate the posterior of model parameters theta. So it's the probability of model parameters theta given the data D, and we are going to access that via the likelihood, which is the likelihood of the data given the parameters, and the prior distribution that just represents any previous knowledge we might have about these parameters. And most of the time, I'm going to assume that that's fairly, it's what I would call an uninformed prior, so I don't have much idea about previous parameter values. One of the nice things about having a PDE model is that if you have a sufficiently simple model for the measurement error, then you can access the likelihood through numerical solution of that PDE model. So in particular here, I'm just going to assume that any observations I have are essentially sort of generated through solution of the model with a Gaussian error or a Gaussian random variable added. Okay, so that's the setup we're interested in. And I've got lots of different data sets, and the kinds of questions that I'm really interested in asking are ones that are really geared around this concept of whether my model is at the right level for the data. And I think that's something that we've kind of, we've thought about the building models at the right level of complexity for a long time. If my model's too complicated, I can fit anything. If it's too simple, I don't learn anything. But we're going to try and hone in more on that, thinking about what the data adds to that question. And so really I'm interested in, can I identify my parameters given the data? I'll pin down what I mean about identifiability in a minute. Can I estimate my model parameters, and can I quantify, because I'm using a Bayesian framework, the uncertainty I've got in those estimates? And I think the real, one of often the most important reasons for being able to quantify uncertainty in parameter estimates is to think about how that uncertainty impacts future predictions you make with your model. And we are going to see some, I think quite clearly that sometimes changing the experimental setup that we have, even if it's only just slightly, can have quite an impact on identifiability and then on predictions. And hopefully we're going to do this at the same time as learning something about the biology. So two of the main data sets I'm going to focus on today are published, one of them in eLife and the other one in Biophysical Journal, and they're in vitro two-dimensional assays that utilize this feature reporter. So in the top assay published by Heinrich et al. In eLife, essentially this is a barrier assay. So you initialize your cell population within a barrier that looks like maybe a cookie cutter. At time 0 hours, you sort of try and carefully remove that. And then you look as the population of cells essentially migrates into and proliferates to fill the surrounding space. And what you can see here is that you begin to see sort of spatial structure building up over time as the population expands here over a period of 40 hours. And in the bottom data set, this is data from what we describe often as the scratch assay. So you grow cells in a dish to a sort of an approximately uniform density of your choosing, and then at some point you take a tool like a pipette or a sharp tool and you scrape away a portion of the cell population. So this is this black space in the middle of this image here. And again, you watch as the cells move and they proliferate and they fill in the available space. And so I want to know, given data sets like this, what can we learn about regulation of the cell cycle through a combination of model and data? I'm going to start, first of all, by talking about this scratch assay, which is the bottom set of those images. And so one thing that's quite nice often with the scratch assay is that if we've got a dish that looks something like this and we make a large scratch, we often only take an image of the kind of the center of the wound field. And what that means is that the data sort of becomes pseudo one-dimensional. We can average in the Y direction to think about sort of simpler one-dimensional mathematical models. So my models are all going to be one-dimensional, and they are going to have very simple descriptions of the cell cycle in terms of cells progressing through being red in G1, green in SG2M, and then dividing at the end of the second phase. So here's what the data look like if you effectively average in the Y direction to get this pseudo one-dimensional data. So when I think about building a model of this kind of system, in particular, sort of building PDE models, then I'm very much interested in asking questions about whether I might be able to estimate parameters of these models given the data. And I think there's two ways to think about this. One is think about a property called structural identifiability, and the other one is practical identifiability. So I want to talk for a little bit about notions of structural and practical identifiability and what they might mean in the context of these models. So here's the question: what kinds of parameters of a model can I hope to be able to estimate from these kinds of data? And I'm going to access it first through thinking about structural identifiability. So structural identifiability is actually something that's a property of the model, not of the data, in fact. And it really just asks the question, if I just take distinct parameter values or parameter sets from my model, did they give rise to distinct distributions of observations? And the reason that that's important is essentially if I want to estimate my model parameters with given perfect data, I need that, right? So if I take two distinct sets of parameter values and they give rise always to distinct sets of distributions of observations, in principle, I should be able to estimate my model parameters from perfect input-output data. And if that's not the case, then it doesn't matter what data I have, essentially, I can't do that. So although it doesn't take into account the limitations that you get from kind of quality of the data, and we all know that in biology that's often a big problem, I think it's still a very relevant first question to ask. So there's been lots of work geared towards developing methods to think about characterizing structural identifiability for ODE models, but for much less for PDE models. So the first thing I'm going to tell you about is a little bit about how we've been making progress in that direction. To do that, though, it's quite useful to kind of showcase kind of how at least one approach to kind of structural identifiability proceeds for ODE models. So I'm not particularly in this particular context interested in the model, but what I've just written down here is a very, very simple model that people have used to describe glucose insulin regulation. So we've got two state variables, Q1 and Q2. They're functions of time, and they evolve according to these ODEs. So I have got what am I trying to say? I've got a control input here called U. So U is something that I can specify. It's an input. It's a function of time. And then within my model, I've got 4 parameter values within the model itself, P1, 2, 3, and 4. And then what I'm going to assume is in this model, the only thing I can measure is Q1 divided by another parameter, which is known as VP. Okay, and so my question is, if I have perfect data for Y, can I estimate the parameters of my model? One of the ways to get at this is by what I would call as establishing a kind of an input-output relation that essentially involves a higher order differential equation for the quantity Y. So if you do lots of differentiating and substituting, then effectively what you can do is describe all the dynamics of your observed variable in terms of this higher order ODE, where it's got the parameters as the coefficients of that higher order differential equation. And the idea is then that if you have perfect data, that you can estimate these y double dot, y dot, and y. And you know what U and U dot are because they're input variables that you can control. And therefore, effectively, if you have perfect data, you'll be able to read off all the coefficients of this ordinary differential equation. And that gives you identifiable parameter combinations. So in this context, I can get P3 from looking over here at the coefficient of U. If I look at then this coefficient, I can then get P1 because if I know P3, I can get P1 from here. And then if I know P1 and P3, then the only thing, the 2 parameters that I can't get uniquely, so only as a product, are going to be P2 and P4. So this kind of structural identifiability analysis tells me that if what I observe is this quantity Y, then no matter how much data I have and how good the quality of that data is, then the only sets of parameters that I can actually hope to estimate from my data are P1, P3, and the product of P2 and P4. So this approach is quite a standard approach to think about structural identifiability for ODE models, but there hasn't really been much work to date thinking about how to extend this to systems of kind of reaction diffusion vection equations, which are the sorts of equations that we're using day in, day out for spatial models. So we asked, could this be extended? So with a view to thinking about this experimental data and this data, this sort of one-dimensional, if you like, spatial data, we wrote down a very simple model that just looks at the density or the evolution of red cells and green cells in space and time. So here I've got both species diffusing, and then I've got a very, very simple model of the cell cycle where my red cells, as they progress through the cell cycle, turn into green cells. And once my green cells reach the end of the cell cycle, then they divide to give me 2 red cells back again. And so we wanted to ask the question, as a sort of motivating example, if all we can measure from our system is the total cell density, could we hope to pin down the 4 parameters of my model? Okay. And so the aim that we had was to reduce the model to an input-output equation, like in the ODE context, but in terms of our observed variable n. And then we know from the sort of ODE results that coefficients in this equation determine your identifiable parameter combinations. So it's much harder to do this in the context of PDEs than it is in the context of ODEs, but you can do it at least for this equation. So using a sort of more compact notation to write down these higher order derivatives, what I've shown here on the slide is just this input-output equation. So one thing that's important to do is to divide through by a coefficient. So you have at least one of your coefficients that's effectively just one. So you've got a monomial. So we trivially can just recognize that we have that already for this system. And then we can read off the set of coefficients of each of these derivatives, and that gives us our set of identifiable parameters. So just reading off the coefficients is what we've got down here in the curly brackets. So you can see effectively that I've got both the sum and the product of the rate coefficients K1 and K2. So they together would be identifiable. And you've got the sum and the product of the diffusion coefficient. So again, those two parameters, D1 and D2, are identifiable. So in this particular context, we can see that our model will be structurally identifiable. We can hope to estimate all the model parameters just by looking at the total cell density. It turns out, and so this is work really driven by Alex Browning in collaboration with a summer student, Maria, and Carlos in the group, that more generally, so if you have a coupled system of reaction, diffusion, and vection equations that are linear in the variables that you observe, then you can always establish an input-output equation. That's like the real battle here. And then once you can establish the input-output equation, then you can essentially establish the structural identifiability. And so if you want to read more about it, there's reference just on the slide, and there's some code as well to help you do that. So the real challenge is deriving these input-output equations. So we can determine structural identifiability of our models. So once you've established whether in theory you should be able to estimate parameters of your model with perfect input-output data, then the next question can become one of practical identifiability. So this is a much less well-defined thing. But really what I'm asking here is it possible to give reasonably precise, and you can put a firm handle on that if you want, so reasonably precise estimates of your model parameters using the data that you actually have available to you, which is generally, well, it's finite and noisy and maybe missing values. So there are some different approaches to thinking about how to get practical or to establish practical identifiability. The first one is effectively to estimate the posterior distribution of your model parameters and then look at the qualities of that posterior distribution by looking at the union, the bivariate, for example, marginal distributions. And for the kinds of models that we are looking at, so PDE plus Gaussian noise, you can do things like sample the posterior using an MCMC algorithm. And non-identifiability can be diagnosed by looking at the MCMC chains that you have, or by, like I said, characterizing the quality of the posterior. How broad is it? Are there correlations between parameters? But the other thing you can do is to use a method known as or method of profile likelihoods. So effectively, what this does is looks at the normalized likelihood of the data, I guess, given the model parameters, given the way this is written. And the idea is that what you want to do is within your likelihood, separate your parameters out into a parameter that you're interested in studying the identifiability of, and the rest of your parameters, and you just call those sort of nuisance parameters. And the idea is to scan over lots of different values, a grid of the values of the parameter that you're interested in, and to essentially maximize the likelihood for over all of the rest of these nuisance parameters. So that gives you effectively a plot of what the profile of the likelihood looks like as you vary that parameter. And then non-identifiability can be characterized by seeing things like flat likelihood profiles. And here's where you can begin to put a firm notion of identifiability, because you can define confidence intervals. So one of the questions we had was kind of, what does kind of what's a relevant level of complexity for the data that I've been showing you, judged by asking questions about the practical identifiability of my model. And in particular for this data set, we wanted to ask the question as to whether the data is sufficient to determine whether cells move with a different speed in G1 versus SG2M phase of the cell cycle. And so to do this, we wrote down two different models. So the first model assumed that cells have different speeds in different phases of the cell cycle. So my cells are essentially red cells, green cells. There's different diffusion coefficients according to whether they're red or they're green. And then we have terms that represent progression through the cell cycle. So we've got rate parameters KR and KG in here, which characterize rate of progression. And then we have a notion of crowding by introducing a logistic growth-like term, which essentially halts progression through the cell cycle as the density moves towards confluence. So that's model number one, and model number two looks almost identical to model number one, apart from the fact that in model two, we assume that the cell, whether they're red or green, so in different phases of the cell cycle, have the same diffusion coefficient. And again, we just assume observation error is normally distributed with a known variance parameter, and we want to establish, is model one more appropriate for this data that I have, or should I be sticking with the slightly simpler model two? Okay, so this is a kind of quite busy slide that just shows you the results of conducting a sort of an MCMC or using MCMC to establish a posterior distribution. So you can see that for each of the four parameters in my model, I've showed you a snapshot of one of the Markov chains. And on the diagonal, I've shown the univariate posterior distribution. So this is the marginal posterior for the diffusion coefficient for the red cells, for the green cells, and for the two rate coefficients. And then you can see kind of on the subdiagonal, they're the bivariate marginal distributions. So just looking at them, the thing that I would conclude is essentially that this model isn't practically identifiable because the chains for DR and the DG, they really never settle down, right? They don't converge. And I have these quite wide posterior distributions for both the diffusion coefficients of the red and the green cells. So I would say that this model isn't practically identifiable. I can't confidently estimate all the parameters, and in particular, it's the diffusion coefficients that I can't get a handle on. This doesn't mean that the model, however, isn't quite capable of predicting the data well, right? So if I do a posterior predictive check by sampling lots of times from the posterior distribution, simulating the model with those parameters and looking at the range of outputs, which is kind of shown in green shading, you can see at the bottom, I can still capture the dynamics over time. So the model itself is fine, but I'm really not confident of the parameter estimates that I have. On the other hand, if you look at model two, which just assumed that the cells have the same diffusion coefficient, whether they're in early stages or later stages of cell cycle, you can see that things change. So the chains for all three of the model parameters settle down very nice and quickly. Everything converges nicely, and you get sort of quite tight posterior distributions for each of your model parameters. So these are marginal posterior distributions. You know, despite the fact that the model is sort of simplified compared to the previous model, when you do this posterior predictive check, you still do a pretty good job at predicting the dynamics of that we're seeing in the data. So I'd say that model two is practically identifiable. And in this context, these MCMC results would lead me to conclude that model two is really the one that I should be using here, because I don't have enough confidence in my parameter estimates in model one to be then confident in making kind of further predictions in unknown scenarios. So I'd use practical identifiability analysis to select model two. The only reason I don't particularly like, if you like, doing this is that this is kind of like taking a bit of a sledgehammer approach, in the sense that it can take a while to generate these MCMC to use the MTMC samplers to generate these posterior distributions, particularly as the complexity of your model increases. So I think it's also, a very, well, maybe a sensible first idea is to actually go and look at these profile likelihoods in order to try and say something about practical identifiability in a much cheaper way. So these profile likelihoods, which are, I'm going to demonstrate, show you almost exactly the same thing and allow you to draw the same conclusions, were generated at a fraction of the cost of those full posterior distributions. So what do they show you? So this is the profile likelihood for one of the rate coefficients, the rate of transitioning from red to green in the model. And you can see that the likelihood, the normalized likelihood, is very nicely sort of tightly peaked around a given value. I've got a closed confidence interval, and I'd say that I can confidently estimate that model parameter. And it's the same for model 1 as for model 2. For all of the rate coefficients, I have these really nicely sort of peaked profiles in my likelihood, and I'd be happy with identifying those model parameters. And you can really see the difference in identifiability between model 1 and model 2 when it comes to thinking about the diffusion coefficients. So again, the profile likelihood for the diffusion coefficient in model 2 is nicely peaked. The confidence interval is closed. We believe that we can estimate that parameter quite well. Whereas if you look at the profiles for the diffusion coefficients in model 1, so assuming they're different, then this is the profile for the cells, the parameter dg, and this is the profile for the parameter dr. We can see that effectively that simply isn't the case. The profiles are much wider, have half open confidence intervals, and I really wouldn't sort of be able to be confident in trusting estimates of those model parameters. So take-home message really is that profile likelihood analysis is something that's much cheaper computationally to perform, but really gives you very similar information to the MCMC analysis. So we might prefer to think about using, or we might think about using profile likelihood analysis as a means to sort of determine the relevant level of complexity of our model in advance of doing lots of analysis of the model, or even estimating the full posterior distribution. Okay, so what have we learned from this talk? Part of the talk, so both of these models are structurally identifiable, in fact, but model 1 that thinks about cells as moving differently or at different rates in different phases of the cell cycle isn't identifiable given the data that we have, but model 2 is, so I should perhaps prefer that model. Really the take-home message, and it's going to come back again, I think, in the talk, is to consider the limitations of the data in building the model, and maybe to use identifiability analysis and profile likelihoods during the model building process. Okay, so we did this work a little while ago, and I think one day I suggested to Carlos Falco, who's in the group, that he might want to take that model sort of off the shelf and just look at calibrating it to this other data set that's published in eLife, with a view to thinking about learning a bit more about mechanical regulation of the cell cycle, or crowding-driven regulation of the cell cycle. And so these are an incredibly rich data set that I'm going to highlight to you now that we haven't mined in its entirety for information. But just to say that one of the things that we could do here is to locate every individual cell in the assay and determine which phase of the cell cycle that it's in. But what we did was to kind of, if you like, reduce the dimensionality of that data by essentially characterizing a density profile for cells in G1, in SG2M, and I guess immediately post-mitotic as a function of distance from the center of this assay. So the kind of data that we have, again, is similar. It's one-dimensional density profile for the different phases of the cell cycle. Now, one of the first things that Carlos noticed when he had tried to apply the previous model to this data set was that it just didn't fit. So this real spatial structure that's very apparent in this experimental data just couldn't be predicted using the previous model that we had. So it became fairly apparent quite quickly that we didn't only need sort of regulation of transitions from SG to M back into G1, so the kind of so regulation of actual sort of mitosis and proliferation. We also needed some feedback of the local cell density onto the rate of transitions from cells in G1 into SG2M of the cell cycle. So essentially what we did was try to capture in general form the sort of crowding based on mechanical regulation of transitions by incorporating terms that depend on the total cell density in our model. So this is kind of very similar model to before, but we've just added the crowding effects in both transitions. And to start with, we looked at just the simplest possible form for the feedback, where effectively they just look like logistic-like terms, where transitions decrease linearly with the total cell density and become zero here at a level capital K1 and a level capital K2. So we have now got 5 parameters in the model. We've got a diffusion coefficient. We've got the sort of transition rates K1 and K2, lowercase K1 and K2, which are characterizing in low density environments, the rate of cell cycling. And then we've got capital K1 and capital K2, which capture the extent to which crowding impacts the cell cycle. So the take-home message from this slide is that if you use an MCMC sampler to estimate the posterior distribution, what you'll see is that the model is practically identifiable. So I can get confident estimates of all the parameters in the model using that data that I showed you. And so we're showing you here the marginal posterior for the diffusion coefficient, for the rate constants, and for the crowding terms. And you can see as well at the top that the model does a pretty good job at capturing the dynamics of the assay, at least right at the front of the invading cell population. One of the things that I think is quite important to notice here is that the fact that the parameter capital K2 is larger than the parameter capital K1 tells you that there's sort of the impact of crowding on transitions from the first part of the cell cycle to the second part is stronger than the impact of crowding on the second transition. So in other words, thinking about this as a cell cycle that has 2 checkpoints that are density regulated in it, the first checkpoint is a stronger checkpoint than the second checkpoint, which maybe makes sense from the perspective of thinking about cells in the sort of SG2M phase of the cell cycle, sort of generating all the new material, rounding up and actually undergoing division. This model says cells only do that if the conditions are favorable in advance. One of the things that you can do then, though, with those posterior distributions is predict then how the length of these different cell cycle phases depends on the local cell density. And so the green and the red just give you predictions based on the posterior maximum, and then the gray just gives you the range of predictions you might see based on the posterior distribution. And you can see again here that this first checkpoint is a stronger checkpoint than the second checkpoint. And that allows you, if you combine all of this, to think about how the total cell cycle length depends on the local cell density, and this is a quantitative prediction. One of the things to say is that a priori, we knew that there was evidence for one of these checkpoints, but not for the other. So when we kind of went into this work, it was sort of making the prediction that both of these checkpoints must exist. But Carlos did a lot of sort of a good literature search and actually managed to show that, or found that in a very recent paper published in 2022, there was evidence for the second mechanical checkpoint in there as well. So the predictions also have some biological sort of basis to them. Other things that you can do with this model is a standard traveling wave analysis, and you can show, or you can make a prediction for the wave speed, and you can show that effectively that doesn't depend on these crowding parameters, but only on the rate coefficients, essentially because this is all based at the front of the wave where the extent of crowding is minimal. And you can conduct a suite of numerical simulations, and that they will show you that, in fact, the sort of profile of your invading wave in terms of cells in each portion of the cell cycle is very heavily dependent on these two sort of crowding parameters, capital K1 and capital K2. That's all in the paper. One of the things that we then sought to think about was sort of reconciling this model and its predictions with the other model that we built and with other experimental data that's out there in the literature. So the top set of data here is data that was actually published in that paper I referenced just a minute ago by Stryker et al. In 2014. And what you see in this assay is something quite different to what we saw in our initial data, which is what's down here at the bottom. And as a result, the kind of invading profile, sort of the invasion profiles of the waves also essentially over quite reasonable time periods look quite different. And actually what's going on in here, in fact, is that, so this is actually also data collected from one of these barrier assays, is that inside this barrier, kind of by time t0, when the barrier was removed, the density inside that barrier is so high that all the cells are contact inhibited. So they're all sitting in G1 phase of the cell cycle. And even after the barrier is removed and you wait for sort of 15 hours, there's very few cells that have actually managed to transition into G2 phase of the cell cycle. And it's only after about 30 hours or so that you begin to see a kind of a rim of cells that are in fact in SG2M in this assay. So these kind of initial conditions in here and the experimental design has quite an impact on the patterns that you see and also potentially on what you might infer. In particular, the model that I just showcased for the barrier assay data was quite different from the original model that we used, and I said that we had to augment that model in order to get it to for it to be able to replicate the data that I showed down here. And we began to ask the question why that was the case. Well, when you delve back into it and you look at things, what was actually really going on, I think, here was in this original data set, first published by Sean Vitadello in 2018, actually the cells are at a really quite a low density initially in the assay. So we've got normalized cell densities on the order of kind of, like 0.5, 0.1. And even 48 hours later, we're really only looking at a cell density at that level of kind of 0.15. And when we used this model or applied this model, I'm going to skip back a bit, for which I apologize, through these slides. One of the things that we swept under the hood, actually, oh, sorry, now I've gone too far. One of the things that we swept under the hood by essentially non-dimensionalizing the cell density to lie between 0 and 1 was estimation of that carrying capacity parameter. So the fact that we could fit the model to this original data set and make progress was really all to do with the fact that the crowding and the regulation of the cell cycle by sort of local cell density wasn't really playing a role here. And in fact, if you just take a model in which there's no crowding-based regulation of the cell cycle and you attempt to fit it to that data, you can see that it very well predicts the dynamics. So essentially, there really wasn't sufficient information in this original data set to pick up the effects of that crowding. Okay, so the take-home message, I think, for this talk is that if we've got quantitative data, that allows us to do so much more when it comes to thinking about the level of model that's appropriate for the data and to really quantify for example, the extent to which regulation of the cell cycle is density dependent. And what we showed in this work is that using the Spuccia reporter with these two different colors enables us to demonstrate that there are at least two checkpoints that are mechanically or crowding regulated based on cell-cell crowding. One of the other things I really want to kind of hammer home is that experimental design matters a lot. So it's really important to consider the limitations of the data in building the model and to be careful of kind of the inferences you draw from the model. So in here, all we've done is sort of rescaled the model so that the cell density went from 0 to 1. It was non-dimensionalized, and we missed a big trick when it came to thinking about what the data could tell us. And the other thing is to say that you can really often learn new things by thinking about taking existing or new models and looking at their applicability to old data sets and vice versa. So with that, I'm pretty much done. Just want to thank members of my group and the people who contributed to this work. Lots of the structural identifiability was really led by Alex. And then Carlos did the portion of the work that's all related to sort of quantification of cell cycle via cell-cell crowding, and many other people, including Matt Simpson and Daniel Cohen, who contributed in terms of data and expertise in the profile likelihoods aspects of this. So with that, I'll say thanks for listening, and I'm happy to take any questions.